It is interesting to know that every second in our body, millions of cells die, and at the same time, millions of cells are regenerated. This regeneration or reproduction of cells occurs by what we term as cell division. So cell division is the process by which a single parent cell gives rise to daughter cells, which may be 2 in number or 4 in number. So according to that, our cell division is divided into 2 parts, which are mitosis, in which one parent cell gives rise to 2 daughter cells. And the another is meiosis, in which one parent cell gives rise to 4 daughter cells. Today, our focus will be on mitosis. So, what is mitosis? As I said you, mitosis in simple term is one kind of cell division in which one parent cell will divide itself into 2 daughter cells. And this occurs only in the somatic cells of the body. What is somatic cells? Somatic cells are all the cells in our body in which we exclude the reproductive cells. Okay, so mitosis is a cell division in which one parent cell divides into 2 and it occurs only in the somatic cells of the body. Another important characteristic of mitosis would be it occurs throughout your lifetime. That means, on the contrary, meiosis will only occur during the reproductive period of your life. However, mitosis will occur throughout the body. So, today we will be discussing about mitosis and its stages. In mitosis, the same chromosome number is maintained at each cell division. This means that at every cell division, the 23 pair of chromosome will be the same. In parent cell will also attain 23 pairs of chromosome, and the daughter cells which will be produced, they will also individually get their 23 pairs of chromosomes. So there will be no splitting of the 23 pairs of chromosomes. So now we will be talking about mitosis and its phases. Like how will one parent cell give rise to 2 daughter cells. So for that we will be studying mitosis under 2 heads, karyokinesis and cytokinesis. Karyokinesis will deal with all the nuclear changes that will take place. That is, first the nucleus will divide into 2 and then after that it will be followed by cytokinesis, in which ultimately the entire cells will be divided into 2 daughter cells. Now look at each phases carefully. This is very important, okay? So, karyokinesis, which is also known as nuclear division, they will be studied under 4 heads: prophase, metaphase, anaphase and telophase. First one is the prophase. Prophase will be studying under early prophase and late prophase. In early prophase, there are 3 changes that have occurred. Just look at it. This chromatin threads, they condense itself to form chromosomes. First change. Second change, these centrioles, they are split into 2 and they are moving far away from each other. However, they are connected via spindle fibers. The third change which will occur is that this nucleolus which you are seeing, it has disappeared in the early prophase. There is no occurrence of nucleolus. These are the 3 changes that have occurred in early prophase. In the late prophase, we will see that these centrioles which were moving away from each other, they have reached 2 different poles. They are now lying opposite to each other. The nuclear membrane, it has also disappeared. And the third change which has occurred is these chromosomes, they have got attached to the spindle fibers via means of centromere. So these are the changes that you will be evaluating in prophase. Followed by the prophase comes the metaphase. In metaphase, the chromosomes align themselves in a straight line in the equator, which is also known as the equatorial plane. This is the signature step to identify metaphase, in which the chromosomes have lined up in the equatorial plane. Followed by metaphase is the anaphase. Anaphase will show 2 stages, early anaphase and late anaphase. In early anaphase, we will notice that the centromere, which was connecting the 2 sister chromatids, the centromere will divide itself. Due to the division of the centromere, the two sister chromatids, they are repelled away from each other. As a result, the two sister chromatids will move away from each other. That will observe in the early anaphase. And in the late anaphase, these two sister chromatids will reach towards the poles, very near to the centrioles, and a slight furrow will start beginning in the cell. This is the initial, or this indicates the beginning of the cytokinesis or the division of the cell. The last phase of karyokinesis is telophase. In telophase, the chromosomes finally reach towards the poles. There is an occurrence of two nucleus. The two nucleus get back all of its components, the nuclear membrane, the nucleolus, and one signature step. The chromosomes have reverted back to its coiled thread-like structure, which is known as chromatin fibers. So this occurs in telophase. Immediately following telophase, there is an occurrence of cytokinesis. In cytokinesis, the furrow will deepen to form two daughter cells, and the two daughter cells will get each of their components, and this ends mitosis, from which we brought two identical daughter cells from the single parent cell.